Hello, listeners. Yamina here. I'm very excited to have you join us for another Dr. GPCR podcast episode. If you haven't heard, we are officially a nonprofit 501c3 organization. Becoming a nonprofit only solidifies our mission to serve the GPCR community. Therefore, we're taking a short podcast break starting in July to come back with new episodes at the end of August, as well as an amazing set of events in the fall. If you are an ecosystem member, by now you know that every Thursday you get your weekly news or weekly GPCR news delivered directly in your inbox. If you're not an ecosystem member yet, join us today. Thanks to our fantastic team of volunteers of GPCR scientists, a GPCR article will also hit your inbox weekly on Tuesdays. Also, mark your calendars for not one, but two Dr. GPCR University classes with Dr. Terry Kanakin. The first one is titled Techniques for Effective Lead Optimization of Candidate Molecules, Principles of Pharmacology for Drug Discovery, which will start on Thursdays, September 5th at 10 a.m. EST and run for 4 consecutive weeks for 90 minutes. Then, on October 3rd, an advanced class will start about therapeutic translation of GPCR activity, advanced method for the optimization of candidate selection. This class will run for 5 consecutive weeks, also from 10 a.m. Eastern Time on Thursdays. For both courses, we will be offering 5 scholarships per course to GPCR scientists who live and work in developing countries. So stay tuned. Just a short week after the beginning of our second Dr. GPCR University class in the fall with Dr. Kanakin, on October 11th, we will be hosting our Dr. GPCR Symposium on GPCRs as therapeutic targets. During that day, we'll have talks from speakers of all levels, both from industry and academia, a poster session, as well as a panel discussion. We're also excited to be co-organizing our very first in-person Adhesion GPCR workshop in collaboration with Dr. Anthony Boucard from the Adhesion GPCR Consortium. Mark your calendars for October 23 to 25 and join us in Mexico City for a fantastic meeting on all fascinating aspects of Adhesion GPCRs and more. If you'd like to sponsor the Adhesion GPCR workshop, please email us at hello@drgpcr.com. Both emails, actually, we have now 2 emails, hello@drgpcr.com and hello@drgpcr.org. Both of these emails will be active for a foreseeable future to make sure that we don't miss your emails. And now, let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR, and this evening I'm very excited to have with me Dr. Badr Sokrat on the podcast. We've tried to do this. We've had Zoom issues, but we're here. Badr, welcome.
Thank you, Yamina. It's a pleasure to discuss with you today.
I'm very excited to have you on. So let's start at the beginning. Would you please introduce yourself? Tell us who you are and what do you do currently?
So I am currently a postdoctoral associate at the NYU Pain Research Center. I'm working in the team of Dr. Nigel Bonnet. We are working on pain, on inflammation, focusing on GPCRs that affect these conditions and how we can use our knowledge on GPCR signaling and trafficking to better treat pain and inflammation. The main goal, I would say, is to go away from the use of opiates. We're all aware of the opiate crisis in the US, in Canada, all over the world. And our goal would be to move away from these drugs to drugs that maybe have less side effects to patients. Previously, I did my PhD in Montreal in the group of Dr. Michel Bouvier. There, I also obviously worked on GPCRs, and I was focusing on the beta-arrestin protein that is really important for the desensitization, trafficking of GPCRs, but also has many other functions that maybe we will discuss later on.
Of course. That was going to be my next comment/question, is that before we hit record, we had a quite pleasant conversation in French, which I have to say, I don't do that very often now, to talk to people in French. So it was really nice to chat with you. And we also talked about our, commonality around, Montreal and working with Michel. So tell us a little bit more about your work, not your work, but about your position as a PhD student in Michel's lab, and how did you actually get into science? That's one of the main questions I'm interested in.
Okay,
I know
so
it's a long.
this is going far away because my interest in science started like as far away in time as I can remember. When I was a child, I remember going to the library to get like National Geographic magazines and like reading books on. My main interest in science actually wasn't biology, wasn't uh, anatomy or anything like that. It was astronomy. I loved the space discovery, reading about the Apollo missions. That was my dream when I was a kid, even a teenager in high school. Uh, in the beginning of every year, teacher would ask, like, "Oh, what do you want to do? Like, what's your dream job?" And for everyone, lawyer, doctor, firefighter, hockey player. Obviously, I'm from Montreal, so everybody wants to play for the Canadians.
Yeah.
Me, it was always I wanted to be an astronaut, and it always like had a few laughs in the room. Like even the teacher would laugh sometimes when I say, "Oh, I want to be an astronaut. I want to go to the moon or to Mars, probably." But that was my dream as a kid, as a teenager, even as a young adult. And so I was always interested in science. I would listen to documentaries. I would read a lot. And when I was getting to the end of my high school years, I was trying to think, okay, if I want to do that, like what road should I take? So obviously, I was thinking maybe go toward engineering, physics. And so I applied to go to College Bois de Boulogne in Montreal, which is a college it's a pretty good college for science. Actually, Michelle went to that same college. We were talking about it.
I did too.
I also would just say, oh, okay, well,
Yeah, yeah,
I
I
guess if
did
you want
too.
to work in GPCRs, you have to go through that
The Bois
college,
de Boulogne.
probably.
Exactly.
So yeah, I went there and we had like everything: physics, chemistry, math. And so during those years, I realized that if I wanted to become an engineer or go into physics, math is very important. Calculus is very important. And in college, I understood that I didn't like calculus, and calculus didn't like me either. So I had to start thinking about other options. I realized I really liked chemistry, and biology was a class that like really I got into it very easily. I was able to follow. I got good grades. I was thinking biology, chemistry, maybe going to biochemistry. And so I applied at the University of Montreal, and I got accepted into the biochemistry program. I did my undergrad there. I did a first internship in the laboratory of Dr. Christian Baron, who works on bacterial secretion systems. I had a really cool summer there working on the interaction between different proteins that form bacterial secretion systems. And they were also working on ways to inhibit the secretion of toxins from bacteria that could cause harm to humans during infections and these kind of things. And after that, the year after this internship, there was every year the biochemistry department was organizing a seminar where different labs at the university would present their research to try to hire masters or interns or PhD students. And so I went to that scientific day, and I was talking to a few of the labs who were presenting there. And one of the labs was present was Michelle's lab, and a postdoc at the time, Bianca Plouffe, was presenting her project. And I didn't know much about GPCRs. Like we had during 3 years of undergrad, I think we heard about GPCRs for like 1 hour, and so it was very, very like on the surface. I knew they existed, but I didn't know much about them. I didn't fully understand her project, but I thought it was pretty interesting, like what could happen down the road
Yeah.
with these kind of studies. And so I was talking to another student during the scientific day about like what labs seem interesting, where I would like to do an internship or to go do my master's there. And I was saying like, oh, the Bouvier lab is pretty interesting. The projects they're working on and the tools they're using seem like they have a lot of possibilities. And he says, if you look behind you, it's Michel. Michel Bouvier is right behind you. At the same time, Michel heard his name and he turned around and we started talking. And I said, oh yeah, I'm finishing my undergrad. I'm looking for an internship for the summer and maybe to do a master's at the end of my undergrad. And so he said, okay, send me an email and with your CV, letter of motivation, and we'll chat. So okay,
Yeah.
I sent him an email, not expecting anything. And if you know Michel, he gets hundreds of emails per day. I wasn't expecting to get a reply, but I was lucky. He replied. We set up a meeting and we set up a potential project I could work on. And that's what we did. I worked during the summer there. with Justine Paradis, who was a PhD student at the time. And it was pretty interesting. We worked on different projects. I was trying to help her on finishing some of her projects. And after that, I had to make a choice, and I decided to stay in Michel's lab for the master's degree, working on a similar project. My goal at the time was to identify beta-arrestin interactors, new beta-arrestin interactors, obviously. And to do so, I used the BioID approach, which is a proteomic approach to identify protein interactors.
Yeah.
And so I did that during my master's. I did also a 4-month internship in Dr. Philippe Wu's lab, where I used more proteomic approaches to look at beta-arrestin interactors. And at the end of the master's, I liked my time in Michel's lab so much, I decided to come back for the PhD.
Love it. I love it. So not only we have Montreal, GPCRs, Brett, Michel Bouvier, University of Montreal, but also Bois de Boulogne in common.
Yeah.
I think that was the college where people went if they wanted to go into science, engineering, med school, and all of those.
Yeah, that's exactly what we discussed with Michel. And actually, I can ask you, how did you find your time in College Bois de Boulogne? Because depending on the person, the reviews are very, very different.
They are. So just to give you, I want to say maybe the first trimester was the hardest.
Okay.
I thought it was hard. I failed my first math test, my first physics test, and my first chemistry test.
Oh.
So then I think I got maybe 39 or 40% results on all of those three, like bang, bang, bang the same week. So not pleasant for a student who was always getting in between 80 and 90% grades. So then the counselors reached out to me and they said, how about you come in and we do some kind of an assessment test to see if you're in the right program. And I did, and they were astonished to see that I was in the right program. They're like, no, you don't qualify, like based on your profile, nothing else fits but where you are right now. So how can we help you? What can you do? And actually, I have to say it's the theater group at Bois de Boulogne that saved my mental energy.
Okay.
Which allowed me to then recenter, refocus, pass the classes that I needed to pass, and excelled at. I loved microbiology. I loved biology. I really, really enjoyed those. I love French literature, which again, interestingly, first couple of exams and the math and physics and chemistry side failed. I also failed the French class, like the entire class. And it was the most devastating experience I never failed a class before. And in that moment, it was the end of the world. But I have to say, I needed to fail that class. I got 55%. So it was on the teacher to allow you to, move forward or not. Yeah. And I had to retake it, and I had another teacher. And that was the best French literature class I've ever had, with the best teacher.
It really depends. The mentor or the teacher can really have a big, big impact on how you take the class.
Exactly.
It's very obvious, right?
Exactly. But it was very difficult. It was very competitive. We've heard about stories of people hiding books that were there's 2 copies at the library back in the day. This was, barely the beginning of the internet era. And it was very competitive.
It's the thing that always people talk about Bois de Boulogne is the competition between students, which there's good and bad to it, but there are extreme cases where it becomes toxic.
Yeah.
And that's something that I don't know if there's any solution to it with the system, how it works right now, the competition to get into med school, because let's admit, a lot of people in Bois de Boulogne at the first semester you ask, and it's like 90% want to go to med school, engineer, med school,
dentist.
Yeah, optometrist also.
Yeah, exactly. I had a lot of colleagues who were very, very competitive, and this was in a good sense, who would, for example, come out of an exam and say, oh, I'm sure I did so horrible, and they would get like 98%.
Oh yeah, always.
And this is the Bois de Boulogne culture, at least in my time. I don't know about you, so let me return
I heard
the question.
like the exact same thing, like all the time. It's exactly the same comment. Like, oh, I did so badly, and then a week later, 98. Or like somebody gets 91, it's like, so pissed because he didn't get 94 because he knows like somebody else who got a better grade. So
Yeah.
it was
Yeah.
very I was lucky in a sense because I knew I didn't want to go into med school. Like since the
Yeah.
beginning, it was not something that I was interested in. I didn't want to go into dentistry. That was not my interest. I knew I wanted to go into maybe engineering, which the grades to go into university weren't as high, so I didn't have that pressure.
Yeah.
But still, you want to have like in high school, I always had the best or second best grade in the class, and I also had the same shock in my first semester. I didn't fail, but my first math class, the teacher gave us a test that didn't count. He just wanted to see the level of the students in the class, and I would say maybe half of the questions I didn't know what he was talking about.
Yeah.
He wanted to see like our level from high school, what we learned, and everything.
Yeah.
And really, like I didn't know where to start to answer some of the questions. And after that, I was talking to other students. Some of them who went to, I went to public school. Some of them went to private school, and oh yeah, we saw this thing that I haven't heard of in high school. And I had a really good math teacher in high school. He was
Yeah.
tough on us. I remember him being really tough on me some days, but obviously he was limited to the amount of things he could teach because
Yeah.
that's life, right?
Yeah.
So that day was like, oh my god, like am I gonna fail this class? But then you adjust, the teacher adjusts to the level of the class. But yeah, the competition at that cégep, that college is pretty intense sometimes.
Yeah. Just to offer some context to our listeners, the not only the college is competitive, it's a really good college, but the system in Quebec is that you complete high school and then you do a 2-year, training or studying at a college before you can go to actual university.
Mhm.
And during this time, you get assigned what they call a cote R, which is this R number, and it's a value that can go, I think, anywhere between low 20s to high 40s, if
Mhm.
not higher. And it's this very, I don't know exactly how they calculate this, but they actually take into account your grades in the class compared to the level of the entire classroom. So if you're in a class where the other students are not everybody is amazing, and you're just above those, then your number, your assigned R number, is influenced by that as well. And that is the number that typically universities, when you apply to the university, take into account. And in each program, and for me it was biochemistry, there you needed to have anywhere between 26 to 30 of that number to enter in the biochemistry program. And that number actually changed based on, so what they would do is that you'd have that number, your tag in your forehead, and then you would apply in the program, and the program at the university would take everybody and they would sort the sheet based on those numbers. And then they would draw a line based on the number of spots they would have. And at med school, you needed to have 35 or 39 above, otherwise you were not even considered. And this is how the universities actually do it. Compared to the US, although I didn't go, I worked as a postdoc in the US, but I didn't go to do undergrad or anything. And here in the US, when I hear people say, oh, you look at your grades, you do volunteering, for example, you write an essay, we didn't have to do that. So that's
it's basically
makes,
how good you are compared to the average of the class decides if you are going to med school or not. And so that incentivizes some people to try to put the other students down to look better, which is, there's no other way to describe it as, it's toxic. It's a toxic way to work,
Exactly. I may have missed this. So after Boulogne, you went to university. What program did you go in? Did you go into biochemistry?
Biochemistry, yeah.
There you go.
I think we had like a very similar
path.
Yeah, exactly.
Yeah. So at the moment, then in the hallway, in the biochemistry hallway, we have Michel's picture with his cohort, my cohort, and your cohort of graduating.
I'm probably somewhere,
students. Awesome. All right. So you mentioned at the moment when you got into studying GPCRs, and that was a little bit later than that, when you started working with Michel in the summer and after that. And you also mentioned that during undergrad, you may have gotten 1 hour worth of, classes on GPCRs.
Mhm.
Once you got into the field, what fascinated you about it? What did you like about it?
I would say the first few weeks were tough because, like everybody who does an internship or a master's degree, the first few weeks you're just reading a lot. And the kind of experiments doing Michel's lab, I had never done before. I had never done cell culture, for example, never done any breath experiment. So it's a lot of learning. So it's a long learning process. And my first task in the lab at that moment was to generate stable cell lines. And that also takes a lot of time before you can start doing your experiments. So for my first month, I was just passaging cells, adding antibiotic, and looking at them for a day or two, counting my cells. And so it was a bit of a boring work. And so I had a lot of time for reading. And so that time kind of played, oh, like there's a lot of GPCRs that are potentially interesting to study. At the time, I didn't know I wouldn't be working on a GPCR during my PhD.
Yeah.
As I said, I was working on beta-arrestin, not a
Yeah.
specific GPCR. But I was like, oh, this GPCR looks interesting. The opioid receptors, there's a lot of potential therapeutic avenues. The chemokine receptors, there's so many of them. They act so differently. So I was thinking maybe I would want to work on this or on that. But yeah, I was just focusing on the on the stable cell line generation.
Yeah.
And one thing led to the other, and I started working on this project to identify interactors of beta-arrestin, and I started doing something that nobody else is doing in the lab. Everybody's doing breath experiment or microscopy, or nobody's doing proteomic approaches. And so that's why we worked with Dr. Philippe Roux, who has a good expertise in these kind of assays. And I found it very interesting because basically when you do this kind of proteomic experiments, you get a long list of potential interactors. And what do you do after that? A lot of literature after that. Go one after the other reading, like, okay, what does this protein do? Does it have any link with beta-arrestin or GPCRs? Has it already been published? And try to identify a few of them that could be interesting to confirm as interactors. And so with the help of Michel, we picked 3 or 4 proteins from that list, and I tried to confirm the interaction of some of them. Actually, it's funny because I picked one that I found interesting, and Michel picked another one that was also seemed that could have a real interaction with beta-arrestin. And I focused mainly on these two. And the one that Michel chose, I could never confirm the interaction. I did breath experiments. I did co-immunoprecipitation, microscopy. Nothing could show the interaction. And the one I picked, I was able to show that. I think you were probably going to ask me like if there's any moments where I had like a light bulb
moment. So this is it.
That was it. It was like, yes, I got it. Like there is a real interaction because when you show it by one approach, a second approach, and a third approach, it's clearly real. And so that moment was a fun moment that like, oh, like I did something right. You know, when you're just starting, you doubt yourself a lot. And when something finally works, you're happy about it. You're like motivated to keep going, you know? Exactly.
Exactly. No, absolutely. And it's interesting that you mentioned let's get back in a bit to the confidence and to the motivation that comes with such successes. But one note I'd like to make is that I actually very much like these assays that we don't necessarily use that much. It depends on the context, obviously, in which you would use them. These multiplexing assays where you can get a lot of data. For example, protein-protein interactions when you're looking for interaction partners, and very quickly you can get that data. And as you did, then you go into the literature, and then pick a couple of those, or if possible, take most of those. Mm-hmm. In order to discover new potential interactors.
Mm-hmm. These are the kind of things that now with the experience I gained, I would have done completely differently. I didn't have the intellectual tools or even the scientific tools to go and study more of these interactors. At the time, I was happy to have one that worked. Now, after a 5-6 year PhD, I'm not disappointed, but I would have liked to maybe pick 20 or 30 or 40 of them. But at the time, I would never have been able to do it. The amount of work it would have required, exactly. I didn't have the tools and like the confidence to go fishing for that whale, you know?
Yeah. No, absolutely. I mean, if you pick 20 or you pick the entire list, then you need to have on the back end an assay that allows you to very quickly sift through those and then focus very quickly. Because otherwise it's just this accumulation of data that may or may not makes sense. But
Mhm.
what I was the point I was trying to get through is that these are the type of experiments where there is a large potential for new discoveries and taking the field into a different direction or
Yeah.
into a new direction.
Mhm. Yeah, because let's say I focus on one interactor, but when the story gets published, there's obviously hundreds more that are going to be available to the scientific community. And maybe
Exactly.
somebody else, maybe a new PhD student in Michelle's lab will look at the list and like, oh, this protein looks interesting. Or somebody in another lab will pick another
Yeah.
one, or like 5 or 10 of them, and goes down that road and discovers another pathway that maybe I overlooked, or that we didn't think of studying. So it gives a lot of information to the scientific community that they can go after.
No, I agree. And I feel like, especially when one difficulty I have, and just because I have a human brain and I'm not a computer, when you have a ton of data, especially these types of assays, these proteomics, this, type of assays generate a lot of data,
Mhm.
and having the tool to try and find relationships
Mhm.
between some of these data points
Yeah.
allows you to gain insight. Maybe you picked one, but maybe in that list, there's multiple that are related somehow.
Mhm. Yeah,
You know?
absolutely. Now there are informatics tools that can be used to
Exactly.
try to map the interaction between different proteins. Maybe the interaction I see is not direct and goes through another one, through another
Exactly.
protein in the list.
Yeah.
So
Yeah.
that's something
Yeah.
that really, like, it's all limited options.
No, exactly. And I think it makes me think about Kathleen Caron's talk at the GPCR retreat last year. And
Mhm.
again, it's serendipity. It's a complete context, but a different in different context. But she gave a talk about Mark Caron, her father, and then her uh, presentation went into the actual work that she's doing. And so basically the aha moment in that presentation was the fact that both her and her dad worked on proteins that are in the same signaling pathway. And he gave her his thesis as a gift, a copy of his thesis as a gift, as a graduation gift for her PhD.
Mhm.
And that's when she saw that actually both of them were working on a cascade at different levels on different proteins, but they were connected by the signaling.
Yeah, that's amazing. Like, it's really like not only in the family, but in the GPCR world,
The
we're
world,
all
yeah,
talking about that, how everybody worked with somebody who knows somebody. We both went through Michelle's lab one way or the other.
yeah,
Michelle and all the people who were in Bob's lab are still collaborating to this day.
yeah,
When you think about that genealogical tree, how big it got today, it's
yeah,
really a big family, and we're all still collaborating with each other. So it's
exactly,
amazing.
exactly. We should actually draw it out.
That
And
would be a big, you need a big sheet.
yes, yes, we can have a virtual one.
Yeah.
All right, so you mentioned working on beta arrestins and not necessarily on GPCRs. And I always ask this question, but what's your favorite GPCR? And what I realize now that it's not the most accurate question, because some people work on G proteins, other people work on arrestins. So here's the question to you: what is your favorite GPCR interactor or GPCR modulator?
So if you ask me today, during my postdoc where I'm working on GPCRs involved in pain and inflammation, I'm really focusing right now on protease receptors and how they trigger or mediate pain in neurons or in the gastrointestinal system. So that's what I'm really working on right now. But it's been 6 months that I started here at NYU. During my PhD, obviously it was beta arrestin. All the multitude of functions it plays in GPCR signaling, trafficking, desensitization. Uh, Obviously, 20, 30, 40 years ago, beta arrestin was seen as a very simple arresting system. Like
Yes.
the name said it all, how it blocks the GPCR signaling. Today, we know it's pretty different, and how important it is for the downstream signaling and intracellular signaling of the GPCRs. And further than that, during my PhD, I worked on non-canonical roles and regulation of GPCRs. That was the title of my thesis. And how, when we talk about beta arrestin, we always mention the phosphorylation of GPCRs as a trigger that
Yeah.
It allows the recruitment of beta-arrestin to the plasma membrane. But I also worked on other post-translational modifications, mainly ubiquitination, which was a big challenge, I would say. It was something not very easy to study, but it was pretty interesting to look at like how important it was compared to phosphorylation, because if you obviously remove this phosphorylation of GPCRs, you impact the recruitment of beta-arrestin. But seeing that the ubiquitination of beta-arrestin or of GPCRs had a very, very big impact also on their function was also very interesting as something that is not very discussed, I feel, right now in the GPCR field.
Yeah, and maybe we should. So let's talk a little bit about your postdoc. And I'm curious to hear, why did you decide to do a postdoc? And how did you go about finding the postdoc lab?
Yeah, how did I do that? When you're doing a PhD, I'm sure you've had the same experience. You don't think about, or at least I didn't, about what to do after that. You're so focused on the next experiment, and I feel like I had that a lot during my 5 years at the PhD, where I was like, okay, what experiments I'm doing next week, and then I realized, wait, I need to start writing my thesis. I need to finish. I need to graduate, and I also need to find what's the next step. And so I knew I wanted to keep working on GPCRs. I knew that luggage of knowledge and skills, I didn't want to just throw it away. So I was looking into GPCR labs all over the world. I also knew I wanted to leave Montreal, not because I didn't like it. I just wanted that experience of living abroad. Actually, I was a bit jealous of other students who did their PhD in Montreal, but we're coming from all over the world, from France, from Brazil, from Sweden. There were people from all over the world studying in Montreal, and I was jealous of that challenge that they took. So I knew if I was going to do a postdoc, I wanted to do it abroad, and so I applied to a few labs that I found were working on interesting aspects of GPCRs. And one thing I really wanted to do, I mentioned that to you earlier, in Michel's lab, I worked on very fundamental aspects of beta-arrestin and GPCR functions. When I was explaining this to friends and family outside of the science world, I always had like a blank look that like, oh, I'm working on the beta-arrestin ubiquitination and its role on GPCR signaling and trafficking. For most people, that means nothing, right? And not only for that, I also wanted for myself to have a project or work on a thematic that was more applied to a disease or a pathology or to a physiological aspect. And right now in Dr. Nigel Bonnett's lab, we're working really focusing on inflammation and pain. And when I mentioned that to friends and family, everybody understands what pain is. Everybody understands inflammation. We've all had Advil, or like, it's something that everybody can understand. And so it's easier to communicate. And also for me, it's the motivation is bigger towards that, because I know that it can have a very significant impact, and not in 15, 20 years. When you're doing fundamental research, that's what you're thinking about, that I might identify a signaling pathway or an interaction or a biased agonist for GPCRs that could be down the road in 10, 15, 20 years, could lead to a therapeutic approach. But right now, what we're looking at is really focused on pain, on inflammation, and how we can fix actual problems from today. And I find that very, very interesting and motivating.
No, I agree. And to my other question, and you mentioned that you sent out the, and I want to get down to the practical aspects. So once you realized, okay, I'm finishing my PhD, I'm writing my thesis, what next? And I want to go abroad, which is, these are very important things. So you as a PhD student, you need to ask yourself these questions as soon as possible.
Yeah.
To figure out, what do you want? I didn't. Again, to your point, focused on the assays, on the experiments. Let's get this out, papers, communicate this, go to conferences, and then we'll see what happens. But at the same time, once you have the answers to these fundamental questions, how did you go about it? You mentioned that you wanted to find a lab where you could work on applicable, more applicable, research towards therapeutic areas, which was in the pain space. But what did you do? Did you start reading papers and trying to find who are the people working on this, or anything that you can share with the audience that could benefit someone who is in your position where you were 6 to 8 months ago?
Yeah, for me, it was pretty easy because I was already working on the GPCR world. So I was familiar with all the names, or what I would call the main characters in the GPCR world. I knew Nigel Bonnett's papers, a few of them I had read already. I had seen poster presentations. I was familiar with him, and I had a friend who did his postdoc here. So that was the first step. For other labs I applied to, I knew them just by reading some of their paper, and it stays in your mind. So when you start applying, you think about it. So what I did is I went back, I selected a few names, and I started reading the recent paper to see what they are working on right now. And the ones I chose to apply for had very, very interesting research thematics. And so I just contacted them out of the blue. In most cases in academia, you will not have an ad saying, "Oh, we're looking for a postdoc." You just have to, like, there are sometimes, but most cases you just have to go and contact the PI and hope that he has a position available. And that worked out for me. And the other thing for me, the city where I was going to work was also important. I wanted to choose a place where I would like to live for a few years at least. Obviously, New York is not the worst city to live in, so I'm happy that it worked out well. So yeah.
Thank you for that. If we can get back to the confidence aspect, when you mentioned that you picked one protein target and then Michelle picked one, and then you were happy that actually at least the one that you picked worked.
Yeah.
And then you had the good, you had a good sense based on the reading, and the reasoning behind you picking that protein actually paid off. I'm curious to hear how much that confidence that you built throughout your PhD helped you go out into the world and get to the postdoc position that you have today.
I think during the PhD, the confidence is a thing that goes up and down. It's not something that you have or you don't have. Yeah. You will have confidence some days, and some other days you'll be lost because for the last 2 weeks nothing worked. Actually, I can tell you the story. On my second or third year of PhD, I had a period of 3 months, and this Michelle doesn't even know that. I never told him about it.
He will if he listens. Actually, he doesn't listen
to the podcast.
So.
Hi, Michelle. During 3 months, I had no experiment give me any significant result, like nothing. And it was not that the results were negative and not like following the hypothesis. It's just that the experiments didn't work for a reason I didn't know. And I was so focused on trying to figure out why it didn't work. I was losing more time than anything. I just had to okay, it's not working. Let's throw everything out and start fresh. And that ended up working again. Something must have been wrong with my reagent or something. And instead of wasting time trying to figure out which one is not working, I just had to throw it all away and start fresh. But to go back to your question, it's something that you need to work on. I know it sounds cliche, but it's the case, because days that you don't have confidence, it's all right, it's fine. Like, go through it and do the next experiment, and if it works, the confidence will come back. Like, I always try to say, and during my PhD, I had the opportunity to supervise many interns and undergrad students. That's something I really liked in Michelle's lab, that I had the opportunity to train a lot of undergrad students. And I always tell them, like, every experiment that doesn't work is just part of the process to take you to the experiment that will work. There's no, actually, it's very, very rare that an experiment will work on the first try. When you're doing your first bread experiment, I always say, transfect this way, read your plate this way, add your agonist this way, try it, but it's not gonna work. Like, I want to give them that, like, to not have their expectation too high. It might not work, it's fine. It's just part of the process, and the next one will, or the third one or the fourth one. So that's part of the process. So when you find yourself down, and everybody will, just, okay, it's part of the process. I'm down today. It's okay. I'll be frustrated. I'll be angry. I'll allow myself to be angry, but I'll move on to the next one, and it's fine. Is that something that you went through during your PhD or postdocs?
Yeah, I think confident, and you're right. It's not something that you're born with. It's something that you build, and I think it comes with experience. And not only good experiences, bad experiences are very important. Those are the moments that, and I don't want to call them bad or, you know, something negative. I think those are lessons learned. And the goal, and it's okay to make mistakes as long as you don't keep on repeating the same mistake over and over and over, and you learn from it.
And confidence is not just about doing experiments. It's also about when you're doing a presentation or you have
everything.
to apply for a job. I remember like the first GPCR retreat I went to, I made a short talk presentation, and I was so surprised to have been selected because like I just started, I have just a few results. How am I supposed to go and present in front of all those GPCR experts? They're gonna like hammer me with questions. And obviously, I had a lot of help from postdocs, from Michelle to prepare the presentation. And so just to have like to accept to do that, because I could have just said, no, I'm not doing this. It's too much pressure or whatever. But it's a good way to have like a challenge. And when you go through it, the next time it just feels normal after that.
You had mentioned, you know, when you mentored students and interns in Michelle's lab, and you were mentioning, okay, you were try to lower their expectation, but at the same time, you tell them you do this like this, you pipette like this, you read like this, blah, blah, blah. I was smiling because when I started my PhD in Nicolas Seveker's lab, I had a summer intern who actually became a master's and a PhD student. And I said to her something that Nicolas loved it. And I told her, do as I say, and everything will be okay. And what I meant by that is I've gone through this. I've made all the mistakes that I needed to make. So when, you know, the angle at which you pipette matters in that. And this became kind of the running gag whenever somebody was interviewing. Nicolas would say, you know, do as I say and everything will be okay. Because of that experience. And that came with the confidence as well. And I think, and it's not only confidence, as you mentioned, doing experiments, it's confidence in general. Having conversations, being able to go up to a PI at a meeting and have a conversation with them. Or, you know, having a conversation at your poster with 10 PIs that come and go, and postdocs, and you don't ever know who's going to come to your poster.
Yeah, how you said, say, do how I say and it will be okay. I had a close one. I had a saying that was pretty close and that also became a gag. It's do it as I say, not how I do it. Like maybe I have a way of doing it that doesn't look the same, but I'm confident enough in doing it that way. But don't do it the same as I do. Do how I say it.
Yeah, my idea was do as I'm showing it to you. And then when you're able to run it, or you're an expert at the way I'm showing it, then you can do pretty much anything you want because you've developed that expertise. But it was do as I say and everything.
Something you said is also true. The confidence will not be only built in the lab or like in your workspace. It will be built outside of it. Like,
yes.
I said something to another intern. Actually, now she's a PhD student in Michelle's lab. And I said she had a presentation to do and she was a lot of stress. And so it was a difficult day. And at the end, when it was done, I asked her, is that the most difficult thing you did this week or this month? And she says like, it's the most difficult thing she did that year. And that was just a simple presentation. And so my advice to her and to anyone, that shouldn't be the case. We should have challenges outside of our workspace, of GPCR, of our lab. And if you have that, what we do in the lab will be pretty easy. Like, and everyone can have different challenges. If you like running and you go running a marathon, that's probably the most difficult thing you will do that year. And going for a presentation or a poster session will feel very, very easy in comparison. Or if you have a big hobby like of building a cabin or something, that would be very challenging. And you will feel that like, doing a bread experiment or testing some antibodies will feel pretty easy in comparison.
And you're right. And I think it's always about the first time you do it. You know, you mentioned building a cabin. Building your first cabin, you're going to make a lot of mistakes. It's going to cost you more than you think.
Yeah.
But building your fifth cabin or third cabin or 10th cabin, you know, the know-how that comes with doing it 5, 10, 20 times will in the long run actually catch you up with whatever mistakes or whatever things you didn't do quite well the first time.
Yeah.
All right, so we're talking about, advice and everything, and you mentioned in the beginning aha moments during your PhD and master's. Any other aha moments that helped you build the confidence or helped you learn something, actually contributed to where you are today?
I think that first GPCR retreat where I was able to do that presentation, that I was feeling very stressed. Obviously, Bob Lefkowitz was in the room, so
I think
it's
I
a
was there.
big deal, right?
Yeah.
It's very nerve-wracking. I remember we were taking the train to go to the conference, and the whole lab we were sitting together, and I just sat by myself because I wanted to practice during the train trip. And so being able to do that just like gives you a little boost of confidence, call it ego, whatever we want to call it, but it does help. And also after that, during the PhD, you will have a lot of challenges, a lot of experiments that won't work, a lot of hypotheses that will fail. And every time you go through it, I feel like it's like, oh, it's fine. It'll be fine. Like the next time it fails, you're not gonna have the same reaction of saying, oh no, life is over. I'm gonna have
End
to
of the
give up my
world.
PhD.
Yeah.
It's always like, okay, it's fine. Like it doesn't work, but it will end up working. Like it gives you like, not everything is gonna be bad forever. Obviously, as I said, when you have 3 months of experiments not working, it's hard to top that.
Yeah.
So after that, like every little trouble or every week that doesn't go as expected, it's okay, it's fine. It's gonna work. I know what to do to go through it. And the thing is, like during the PhD, obviously you learn how to plan experiments, how to manage projects, but you learn how to go through challenges. You learn how to learn. You learn how to go and find answers to your questions, because that's something that's science, right? It's not like something that's already known. You have to go and find new answers, and the most important skill is like to go and be able to find those answers. Nobody will give it to you because we don't know those answers. We don't know how to identify that specific agonist for that orphan GPCR, right? So it's something that you have to go and find by yourself, and that's something that I feel like that's the real goal of a PhD.
Yeah, and I think it also one other thing I want to add to that is, the from a confidence perspective, from also a scientific perspective, I think the PhD and the postdoc are the places or the experiences that you need to have in order to learn things on your own, but at the same time, think about the fact that the goal is to do science as a collaboration and be able to say, here is the design of my project. This is how I see, or I hope, this is my hypothesis. This is how I project that it will go. And this is the limited time that I have. So who is the expert who can help me to get answers to my questions as quickly as possible, so that you can move forward? Either it's answers, either it's a collaboration, either is, thinking about a type of assay that you may not have experience running. And then you have to make the choice of, do I want to learn how to run this? Or do I want to learn how to code? Or do I want to find a collaborator to get us there faster?
Yeah, I'm smiling because I literally had the exact same conversation today at work with a colleague where I was discussing a project I'm working on, or I'm building this project right now. And I had like the plan. I want to do this, this, this. I always like to start the project by imagining what the manuscript would look like. So I'd figure 1, 2, 3, and the type of results, assays I would like to have in each one of them. And for some of those assays I want to do, I want to learn. I want to do more confocal microscopy because I didn't get the experience that much during the PhD. So I want to do those experiments. Other experiments, I don't need to learn because it's simple kits that somebody else is already running. And so I will go and ask that person, hey, are you interested to work with me on that project? I know you're using this kind of assay, you're using this kind of experiment. I could just give you the reagents I'm using and you could run them for me and participate in the project, instead of wasting a lot of time to learn the technique, learn the approach, learn the protocol. And instead, you can just give it to the person. And if I needed to, I could just go to them. They will teach it to me at some point. But you have to choose which assays, experiments you want to learn, which ones you're an expert in and you will do the experiments, and which one you're going to delegate to somebody else, be it a colleague, an undergrad student, an intern. you have to learn how to delegate.
Yeah, I think delegating is also a great point that we brought up. And I don't know about your experience as a PhD and currently now as a postdoc, but back in the day, and maybe it's just me, in my mind, especially in the beginning of my PhD, I thought I had to do it all on my own.
Same. I thought the same.
Right. And it was the last, so I had enough papers after 3 years to graduate. But because you need 2 papers at the University of Montreal, 2 publications to actually start writing your thesis and graduate. But I had started a third project, which wasn't okay. I started it, but it was Nicolaus's idea, basically. He asked a question and I said, this could be interesting. So we started working on that. But that's the project where at some point I said, no, this is not the way we should go. We need to go into a different direction. These are the assays I want to run. And I literally went around in the lab and said, you do radioligand binding on another project. Can you help me with this? You do flow cytometry. Can you help me with this? You do this assay. Can you help me with this?
Yeah.
And not, it's a molecular pharmacology paper. It's CCR2 with its chemokine ligands. I love that paper because one, it allowed me to, put on my big girl pants and say, I need this. We need to finish. I had to finish on the time. I had a timeline. Also, rally the troops around this project.
Yeah.
The data was so beautiful. N equal setup, N equal 3 publication quality figures, which is a miracle in the world.
That never happens.
Exactly. And then the reviews, we had 0 questions that required us to do basically anything. There was one question. We had a table summarizing the pharmacology, and we had the abbreviation ND. And the one question we had is ND not detectable or not done? And I actually printed out the reviews for this paper because this was the smoothest flying ship ever.
I'm sure the reviews fitted in that post-it note,
Exactly. It was tiny. It was beautiful. Everybody was commenting positively. There was nothing.
But that goes back to the confidence we were discussing earlier, to have the confidence to go to your boss and say, hey, this is how I want to do it. And then the confidence to go to your colleagues, to your collaborators, and ask directly, like, hey, can you do this? Can you do that? That requires confidence that I don't think you would have had your first year during the PhD.
Definitely not.
Yeah.
Definitely not. Because during your master's, you're focused on pipetting and learning how to pipette and not to, pour an agarose gel with water, made with water.
Yeah.
That's
I made the mistake. I made that mistake.
I did that too. Until you load your samples and you see them diffuse and you're like, what is going on? And then towards the end of your PhD, you get there. And then in my postdoc, I did similar things at Rockefeller where I proposed a project, and during the discussion, so I proposed to look at heterodimerization between CXCR7 and CXCR3. Because of the relationship between the ligands and potentially the co-expression of these receptors on immune cells, for example. And so during the discussion with Tom said, well, wait a minute, the CXCR3 has multiple chemokine ligands. What do we know about this? And it also has different splice variants. What do we know about this? So I took that question and developed it into this story where I took the known chemokine ligands, there's 3 of them. I had the splice variants synthesized because you can buy them at the time from cdna.org. And by trying to answer this question, at the time when I joined the lab, there was just calcium assay performed in the lab. By the time I left, we were doing in-plate Western ERK phosphorylation. We were doing bread assays, G protein, receptor bread assays, beta you name it. We did it. And developing these techniques allowed me also to collaborate within and outside the lab. And it was, in a way, I didn't plan it from day one when I joined Tom's lab. It's just that as I was building out the CXCR3 story, I realized, oh, I need this tool, I need this tool, I need this tool. And then I went to conferences and people were like, oh, you have that, let's collaborate. One of those collaborations, I think, is finally coming out. The fruits of that collaboration is coming soon out. And I met someone at a conference, and I tell this story a lot of times on the podcast. We ended up collaborating, and the data that I generated in 2015 is included in the paper that we submitted to Science. There's about 70 people on the paper at this point, because we started out with GLFI2 mutations that are occurring in a patient population.
Mm-hmm.
And so there was this entire long study to understand how does that mutation, that single point mutation, and it's not in one residue, it can be in multiple residues in the G protein, and it escapes me where exactly. But that actually induces a lot of immunological problems to patients. And it's a small patient population, but the problems are widespread. Mm-hmm. So this one conference where somebody introduced me to someone led to a really cool collaboration.
Yeah, this is like how it should be done. And when you were talking about all the different approaches that you developed in your lab, it reminded me of one of the big reasons why I decided to do a postdoc. You asked me that question earlier, and I forgot about that. I wanted to learn new approaches. And so I might ask you one question. During a postdoc, how do you balance that need or want to learn new techniques to kind of have, I knew how to do this, that, and the other, but also the need to publish and publish fast? These are two needs that are not always going in the same direction, right? Because if you want to learn something new, it's going to take you some time. Whereas if you collaborate, it's going to go faster, but then you don't learn that approach. How do you balance these 2 points?
That's a very, very good question. And I'm trying to think about how did I approach that. And it was more organic than anything else. Mm-hmm. At that time, similar to my PhD, I was so excited to live in New York. Again, we have that too in common. And to be at Rockefeller, to be in Tom Sackmar's lab. It was very exciting. And my mind was on lab, data, these chemokine receptors, and getting that. And basically, I don't think similar from the perspective of how I went about the CCR2 project in my PhD. For this particular CXCR3 project, I focus on developing these methods because I tried to look around and I couldn't find anyone who would be willing and able to help.
So you didn't have the choice. You had to learn those approaches.
I had to do it. I had to learn that. I had to develop it. I had to make a case to say we need to spend $1,000 per construct. These are chemokines. Chemokines are expensive. I had to lobby to get 50 micrograms and not 10 micrograms. And I had to consistently show that I'm able to generate data and that it's making sense.
And was that advantageous to your career, learning all these approaches, developing these tools? Or was it just, okay, it was good for this paper, for this story, but did it bring you something extra for the rest of your career?
It brought me the confidence to say I can solve this problem.
Okay.
That's what it brought me. But from an experimental standpoint, once I left Tom's lab, some things I couldn't use anywhere else because those were instrument dependent and we didn't have the instruments in my other lab. But that work on CXCR3 and the methods developed around it allowed me, one, to publish. It took 5 years because of multiple reasons, but allowed me to publish my first and last 2-author paper. That was an uphill battle, but at the same time, it also allowed me to kind of try and answer your question. Once I developed those methods, it allowed me to collaborate. So to increase the number of papers that I was on, because I developed those methods. Yes, it was hard. I used it as a way to get that CXCR3 story out, but it allowed me to collaborate on a CXCR4 story with NIH. It allowed me to collaborate on a CCR5 story. I taught others in the lab how to use it. I may not be on those papers, but that's kind of a legacy there.
So that goes back to the point we were discussing about the confidence, but also that in science, doing a PhD or a postdoc now, my goal shouldn't be to learn new tools. It should be to learn how to learn, learn how to find the answers. Show that my CV shouldn't be just a list of techniques or protocols I know. It should be a list of time I was able to go and find an answer. Like this time I had this challenge, I was able to solve it by doing that. And that's that's how I should be able to build a CV during my postdoc.
I agree, and I think that's important, but it goes also back to what is your goal after your postdoc? You know,
That's the
if
million
you
dollar question, isn't it?
right, exactly. And I'm kind of asking it to you, but before you may or may not know this because you started 6 months ago. One piece of advice I can give you, and anyone who's in their postdoc early on, ask yourself the question right now.
Yeah.
What do you want to do? If you want to go into industry in the private sector, what you just mentioned, your ability to identify a problem that has a significant impact, the ability to figure out what are the tools that you need to try and get your answer, and identify and be able to make the choice of, do I have to learn this, or can I find someone?
Yeah.
That's the problem solving thing, because then you remove the lab, you remove GPCR from that layer, and we can put you in any system, and you can apply the same train of thought to solve a problem. If you are a problem solver, then you can basically go into any field, whether it's in academia or industry, although academia has its own quirks. But in industry likes this. You need to be able to learn quickly. You need to be able to solve problems.
Yeah, that's my main goal during the postdoc. It's yes, I want to learn new skills. I realized that okay, I was very, very good at studying GPCRs, beta arrestin by Brett acid in my PhD.
Yeah,
that wasn't enough, in my opinion, for myself. I wanted to learn something extra, so I came here. I'm gonna be exposed to different approaches. I'm still gonna be able to work on my favorite subject, GPCRs, but I want to be exposed to other ways of solving questions about GPCRs. And by the time I'm done here in New York in a couple of years, I want to be able to say, oh, I was able to solve different problems, different questions, but by using different tools that I didn't have, and I just learned how to do it. And the managing aspect of project is something that every PhD student does that. But now I really want to have that expertise. You're like, okay, I start projects from the beginning, I took them to the end, and I manage the project completely by creating collaborations, by contacting other PIs, by discussing with colleagues in the pain research center. That's something I really like about the setup of the center here at NYU, is that we have every expertise that we might need to study GPCRs or study any pain-related signaling pathway. We have someone that has that expertise that could help us, who could teach me how to do those kind of experiments. And so that was one of the main factors that drove me here to New York.
And it's interesting that you mentioned project management, and that's very, very interesting and very important as well. And part of project management is not just about the techniques and the reagents and everything, but it's also about figuring out what is the equipment you need, who are the people that need to be doing what and when to move the project forward. I see you're smiling.
That's mine because I realized that when I started here, in Michel's lab, we have Maman Monique, that you know, that manages the lab, and that I didn't realize a lot of problems were invisible to us because we had Monique that was kind of solving them in the background. Now that I came here and I had to kind of develop project from the beginning and do my own ordering of reagent and organize the lab in a certain way, that's also a skill that I'm glad I'm learning right now that I didn't have because we were a big, big team. The senior postdocs and managers were kind of helping with that aspect. Now that I'm doing that, I'm like, oh, this is part of the job of a scientist to also kind of take care of these aspects, the reagents, manage the equipment, to maybe have to, if you want to do some experiments that you don't have the proper equipment, and contact the vendors and negotiate. Something as simple as buying new multi-channel pipettes for the lab and having to discuss that with the vendor from the company and kind of negotiate to get free tips. It seems like nothing, but it's part of the skills you have to learn during your postdoc, or if you go if you stay in academia,
No, I agree, and then now my turn to smile, because in my mind that's the That's where you need an amazing lab manager. And it depends on obviously the setup you're in. But if I were to build my dream lab, doesn't matter if it's academia or industry, first things first, you need a fantastic, well-organized, detail-oriented lab manager.
Yeah.
Because in the long run, you do not want your scientists to deal with these things.
Mm-hmm.
You want the scientists to do science. Depending on what level the scientist, this could be a research associate, like a technician, or a senior scientist, or even principal scientist. Everybody has, in the big machinery of discovery, whether it's fundamental work or drug discovery, has their role. Because what we do in general in science is very difficult. And in order to maximize a positive outcome, a successful outcome, that be a drug or a paper, two very different things, I realized there needs to be a well-oiled machinery, a system where everybody knows what their circle of work is and how do they fit in that big machinery. Now, that doesn't mean that you're restricted in your own box, but if you have a 30,000 feet view of how that works, then you can say, okay, this is the role, not necessarily the person, but this is the type of role that does this. And this supports the next group this way, so that this next group can deliver this to the following group, and so on and so forth.
Yeah. Because we don't realize, but every human, every scientist has a certain bandwidth of tasks they can accomplish in the day. And some are obviously more important than the other. But having just a small, like, noise in your head saying like, oh, I need to do this, take care of that, that is not really related to your project, your experiment, it does take your brain out of, like, when you're doing or writing the paper or something, having that in the background does take something away from scientific process.
Exactly. And when you're stuck in conversations or emails with vendors, I mean, it can be fun because, you do it once or twice, but when it's the third or fourth, fifth time you have to deal with this, you don't want to do that.
No, I know.
You'd be doing experiments, reading papers, finding collaborators, whatever.
Yeah. No, it does take time after the day. And we have obviously, like, you have your time in the lab, but when you leave the lab, you want to take care of something else, and then you're still in your emails. So obviously, the less of that, what they call administrative work in the lab you do, the more you can focus really on the science.
Exactly. And I'm mindful of the time. I know there's a hockey game on. So I had a question and it did escape me. But let's go with the following one. Any other advice that you think you'd like to share with PhD students, master's students, even postdocs?
The one that comes to mind now is that syndrome we all deal with, is the imposter syndrome. I'm sure you've discussed this. Sometimes we talk about it between us, the PhD level, that you have some data that you have to present and you feel like it's not good enough, or that, like, oh, you want to apply for a job and maybe I'm not good enough to apply for it. And that's something that doesn't go away. Like, it's from the first day when you start your PhD until you're doing your postdoc. And I have some results right now that I want to show my boss. I'm like, is it enough? Is there enough data here? Is there enough to, like, make a story out of it? That doesn't go away. And, like, even before the PhD, when I'm at that scientific day at the biochemistry department, I'm talking with Michel Bouvier, and I'm like, he's a big deal. Can I really, like, ask him to take me in his lab? Like, why would he take me? There's 300 undergrads in the room. That doesn't go away. And it's just, there's no other way of saying it. It's like, it's a choice you make to, like, maybe he will say no, or maybe the paper will be rejected, or maybe the data is not good enough, but maybe it is. Like, it's as simple as that. And if you don't try, again, it sounds very cliche, I know how it sounds. If you don't try, it's obviously not going to work. So if it's like applying for a postdoc or applying for an industry job, if you don't apply, you're not going to get it. We're talking about hockey. It's a big Wayne Gretzky quote: you miss 100% of the shots you don't take, right?
Absolutely.
So
very
try and
love
see
it.
what happens.
I love it. No, I do agree with that. And I think about that a lot of times. And I think imposter syndrome, let me ask you this. Do you think, you mentioned having conversations with PhD students about the imposter syndrome. Do you think, is it the same to the same extent for depending on your level or whether you're a male or a female scientist?
I did hear from female students, female researchers that they believe they deal more with it than men. I don't know if it's true or not. I obviously don't have the data. Is that in that sense that you're asking?
Yeah, exactly. So I think from the perspective, I think there's obviously, in my mind, that's how I think about it. And I have this conversation oftentimes with people and with men, and you go into a room and I think we all have the same fears. We feel the same way. If we have imposter syndrome, we experience it independently of gender. But what we do with it might be different for male to female.
It can be as simple as you're in a conference, in a seminar, and at the end of presentation is the question period. Who's gonna raise their hand to ask the questions? I'm sure there's data on this. In my experience, from what I see, you're right. It's mostly men who are gonna get that. Okay, I have a question. My question, and this is something we all have as scientists. We have that, like, oh, my question is smart. My question is important. So I'm gonna ask it. So the question is, like, you balance that with like the imposter syndrome, or like, no, my question is not good enough. I should not like humiliate myself by asking a stupid question, right? So it's that balance of like, oh, I'm very smart. I have a very good. You always want to ask that question that like everybody's like, oh, this is really good. Oh, how he was thinking about that. But you don't want to be the person asking that question. It's like, he just showed it 2 slides ago. Why are you asking?
Exactly.
So you have to balance both.
I think that's that. I think and I'm speculating completely here, but I think I don't think we're let's say we're at a conference, GPCR retreat, and then somebody finished presenting. Most of the time, let's say men ask questions. I think what goes on is they genuinely have a question and they just ask it. I don't think, I hope not, at least people are like, this is the best question I can ever ask. I need to ask it right now. I think it comes out of this space of, I'm interested in this, I'm going to ask it. For me, especially as a student, it did happen a lot of times where I had a question and I told myself, you can go talk to the person later.
But where does that come from? Is it from fear that the question is not good enough?
Yeah, exactly.
That's exactly, right?
Exactly. It's what will other people say about my question?
Yeah.
And then the most frustrating thing is that somebody else asks the question, the same question, and you're like, dang it, I should have asked that.
Yeah.
You know? So I think it's there. And I think if we set aside, and it's hard, but you have to set aside this, you have to say, I feel the, imposter syndrome, but I'm just choosing not to let it keep me back.
To not listen to it, basically.
Exactly. And one I think what helps is, one thing we haven't touched upon is work-life balance, is having, because we talked a lot about lab work and doing experiments and stuff, but it's important to be able to disconnect. It's important to have that support network, family, friends, pets, you name it. It's important to have activities that are not lab work, science related, so that you can let your brain process in the background.
Yeah, it's funny that we didn't talk about that, but I feel like that was the most important part of my PhD. I had something else. I had basically a second life outside of the lab. I was doing a sport. I was competing. I had workouts every night after work. So I knew, like, I have to leave the lab at a certain time because my coach is waiting for me. I have to, I'm not going to be in the lab Friday, Saturday, Sunday because I'm competing 5 hours away from Montreal. Having that during my PhD helped me a lot. And that goes back to having challenges outside of work. Whereas the most difficult things I did during my PhD were clearly not in the lab. I had way more difficulties and challenges, like in the competitive environment I was in outside the lab, than in the experiments I was doing. So when the experiment failed, it's fine. I'll do it again. If I failed in the competition, I lost. Like it's over. Like I cannot go back. I cannot ask the referee to start over again. It's done.
Yeah. And may I venture that it's boxing based on the image in the background?
Yeah, that was it. Yeah, that was. Which way is it? It's that way.
Yeah, you did it right. Yeah, no, I think that's important. And that's hard too, because you're so wrapped up in your, this trajectory as a scientist, whether no matter what level you're at, and it goes beyond, academia and industry. But this is needed. Like unless there is a stringent deadline and there is something that needs to be done, I never worked on the weekend, and I don't work on the weekends.
Okay.
That's one thing. That doesn't mean that I don't think about work, but I realize that, organizing some stuff outside allows me to be more creative, because while I'm doing that, that's when I have ideas like, oh, we should do this, we should try this, we should do that.
Yeah.
And it's very, very important.
Exactly. And what you said about kind of detaching from work from time to time.
Yeah.
Like obviously with boxing, if I'm going to the gym after work and I'm sparring, I cannot think about my breath acid because the guy in front of me wants to break my nose, right?
Yeah.
If I don't move my head out the way and I'm thinking about the lab, I might not make it to the lab the next day. So this is like a way of like just leaving the lab and doing something else. Be a sport, going for a hike, doing some painting, whatever you're going to the movies.
Recording a podcast.
Doing a podcast. So actually, like if I can ask another question, like how did you get into podcasting? Like how did the idea come?
So the idea came in 2020, and this was January 2020, and I was listening to another podcast. I used to commute to work, so I listened. It was an hour each way, although it was, close enough. And I was listening to a podcast called Consulting Success, and they were talking about how do you differentiate yourself in your field, or in, as a person from everybody else that has the same experience as you do have. And they were saying, well, podcasting, they were talking about, starting a podcast. And I came home and I told my husband, I was like, I should start a podcast. And he looked at me and was like, yeah, but what are you going to talk about? And I said, here's the beauty. I'm going to invite people to talk to. and that's how it started. And I was so paralyzed the first couple of episodes. So the first episode was with Paul Insel, who's at UCSD. And Paul is wonderful. And thank God Paul went into stories and explained and gave context. And so I felt comfortable with him because I knew him. Number two, I didn't have to talk that much either in the beginning. This is episode 1. And the second episode was very different because this was with Thierry Hébert. And Terry asked me questions, and I had to say something. So that was challenging as well. And I think these are the first two episodes. And I think after the first two, I felt more and more comfortable, to the point that in that same year, December 14th, I was recording with Bob Lefkowitz. And I remember feeling very excited to talk to Bob, but I didn't feel stressed out about, oh my God, I'm going to interview Bob Lefkowitz. Obviously, in a bad way. Like, I was very excited, but I was not petrified of, oh, I have to talk to Bob.
Yeah.
No?
So like if you listen to I know the 150th episode is coming soon. If you listen to the first one and then listen to 149th episode, what do you think is the main difference? Or like what's the big upgrade you did? Or like how did you improve as a podcaster in between?
Oh, as me being the interviewer?
Yeah.
I think it's a level of confidence.
Yeah. Okay. Again, same answer.
It's a level of confidence, and you're right. And now, and the one I have one rule is I don't look up. Okay, that's not. If I say I don't go and do a deep background into the guests.
Okay.
Because I want to be able to ask questions that anyone who would meet the guest would be interested. And I don't think I'm that special. So I think what the questions I'm asking are pretty good questions that anyone else would be asking if they were having the same conversation. And that's why each episode is unique, because it also depends on who the guest is.
Yeah.
If somebody wants to stick to the topic, fine, we can go with that. If somebody like you wants to have a conversation and ask me questions and go these with, fine, that goes too. And I think that's what makes it unique.
It's very fluid, it's very flexible, it allows it to go in in different directions.
Exactly.
Yeah.
Exactly. And I can also tell you that what I've noticed, speaking of confidence, is that depending on what setting I am in, whether it's a work setting, whether it's a Dr. GPCR setting, or even within Dr. GPCR, a podcast setting, I realize that my confidence shifts.
Okay.
I have to push myself in in other professional settings to embody this Dr. GPCR. The person that you're talking to,
Mhm.
I have to tell myself, why are you not that? You should be that.
Because now
Sometimes,
do you feel like you represent more than yourself now with the Dr. GPCR name like attached to you now?
yeah, people recognize the name. They don't necessarily recognize my face, but when I say,
Okay.
Dr. GPCR, like, oh, that's you.
Mhm.
Or I've had people who I met at conferences like, oh my God, I cannot believe that I met you. And it's a very humbling thing, you
Mhm.
know? It's just, I think the way I approach the podcast and the Dr. GPCR ecosystem and everything that we do is unique.
Okay. And
That's
where do you want to take like what's the ultimate goal of the podcast and the Dr. GPCR
I
world
love your
now?
questions. I love your questions.
Because I'm really curious to see like what do you want to do with this now? Because
Yeah,
you have
so
unlimited options, right?
yes, absolutely. So from a community and ecosystem perspective, I want Dr. GPCR to be the hub to serve the GPCR community across
Mhm.
the world, independently of where you are, what do you do, what industry, what sector you're in. You're working on GPCRs or related proteins, you're welcome to find resources, to connect, to help. And to do that, well, we're recording now. This episode is going to be released after 151, actually. But as we're recording, as of 2 weeks ago, we are officially a not-for-profit organization,
Mhm.
which was something very important to me because we want to serve the community. And I want to offer tools, networking tools, learning opportunities to everyone in the field. And I have a ton of ideas we haven't even explored for us to do that. I hope that answers your question.
Yeah, I can tell you as a someone who's been working in the GPCR field, and I will continue working in GPCRs. I'm really grateful for this word that you're opening, that we'll be able to create contact by listening to the podcast and hearing what other people are working on. It's gonna be you cannot read every paper that's published on GPCRs, but listening to the podcast once a week and like, oh, I didn't know that this lab is working on that, or having contacts through the GPCR system.
Yeah.
That's going to be a very, very good opportunity for
Yeah.
younger students, for postdocs, for PIs. So this is
Exactly.
a really cool initiative.
Thank you. And I think with the Dr. GPCR University, I think we're taking it to the next level. I don't know if you saw it, but in February we launched the university, starting with a course with Dr. Terry Kanakin about applying pharmacology to drug discovery.
Yeah.
By the time this episode gets released, we will be either in the middle or towards the end of the data analysis hands-on workshop with Dr. Sam Hoare.
Mhm.
And the idea is serving the community. And I'm always welcoming, emails, phone calls, texts, you name it, as to what do you need as a community? What do we need as a community? So far, it's been all coming out of the need of Yamina the postdoc, or Yamina the PhD, or Yamina the scientist, in biotech. Because these things are not trivial. These things are not available anywhere else. You might go to one-day workshop, but then things are not recorded. You might get or may not get slides. And with the university, you're part of a small group. The small group has its own chat or, news feed, basically. You get the recordings, you get the materials, you get access to the teacher. Even after the course is done, you get all sorts of things that you may not get if you're in let's say in university. Like you were mentioning that you're right now in an institute where you have access to all of these opportunities, these tools, these people, but you're lucky, it's not the case for everybody. So we want to decentralize that from a physical place and have that online. And I think COVID taught us that. that this platform and this way of thinking and connecting has its place.
Yeah.
You know, I discovered labs in Central America that work on GPCRs, and we have people from all around the world, and sometimes I see, you know, the countries pop up and I'm like, wow, I didn't even know a lab existed there.
Yeah, no, this would be like create a lot of new opportunities if you're looking for a PhD lab or for a postdoc lab.
Yes.
Or I think for collaboration is going to be amazing. I
Yeah.
don't know how far we're going to go and like the information is going to be available. But if we have access and we know that, oh, this team is working with this approach and I really need this for a project, I think it's going to be the hub where everybody's going to be able to link in a certain way
Yeah.
and collaborate, work together.
The way I think about it in simple terms, it's the GPCR LinkedIn, where you have the university is the GPCR LinkedIn Learning, where you have, you know, the feed, the groups, you name it.
Yeah.
And what I want, my dream is that when people wake up in the morning, I don't know when you have your phone in your hand first time in the morning, what you're looking at, but I want you to go to the ecosystem and see, okay, what's going on? You know, that's my goal.
In my part, like I'm always looking forward to the newsletter with the GPCR news and everything's happening in the last month in the GPCR field. It's always like, oh, okay, like I missed this paper, or like I missed that this happened in the ecosystem, you know?
Yeah, we have that in a weekly basis. The GPCR news, the Dr. GPCR monthly news is more as to what we do. And it's funny because I was so focused up until very recently on getting these things out and getting trying to figure out, okay, what do we put in that weekly news? You need content, you need pertinent content. And how do we get that content? And how does that look? And how do you, it's really all the behind the scenes type of things. I hate to admit it, but I don't read it. Because by the time it's done, you're like, phew, it's got out. And I have to say, I don't physically prepare the thing, the news. I have a team in the background, and episode 150 is actually going to be, or has been, now that if you're listening to this, an introduction to our team of volunteers. Including, and I think she confirmed, our very first Dr. GPCR volunteer who joined us the first time in July of 2020. So I think it's going to be kind of a you know, going from one volunteer to now having 6.
Okay, so a moment of celebration for everyone who worked on this project, right?
Yes, exactly.
Okay,
exactly.
I'm looking forward to listen to it.
Fantastic! I can't wait. We're releasing on May 9th. We're recording actually this weekend in the evening because we have team members in Australia as well. So we're from all across the world. We have people in Europe, we have people in North America, we have people in Australia, and it's challenging, but we always have, you know, really we find the time to talk to each other.
I guess that was the goal for Dr. GPCR, to go global. So
exactly,
we're gonna have to work through time zones and
exactly. So. Super. I don't remember the last question I had for you before we got into this, but if I do, I will let you know. Badr, thank you so much for being here today.
Thank you very much for the invitation. It was a pleasure.
Thank you. Don't go anywhere. I'm going to stop recording.
All right.
I need a favor. Please share the Dr. GPCR University program with your friends and colleagues, especially if they live and work in a developing country. Please subscribe to our YouTube channel. Many of you come back to watch our videos, but aren't subscribed. Having more subscribers will help us get you more content. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest and our Dr. GPCR team, Attila, Ines, Montserrat, Ivana, Cam, John, Yatsu, Andreina, and Balint. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. And don't forget to subscribe to our monthly newsletter on our website to get all the news about all the work that we've been doing and we are doing at Dr. GPCR directly delivered into your inbox. Another great way to support us is to share your favorite Dr. GPCR programs with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com or hello@drgpcr.org. As I mentioned, both emails will be active for a foreseeable future to make sure that we don't miss your email. Until next time, stay safe.